Hey guys, welcome back to my channel. In this video, I'm going to be attempting 40 biology HL past paper 1 questions. Seeing the positive feedback I received for the chemistry version of this video, I decided that I will continue this series where I do past paper 1 questions on biology HL. So before we get started, I'd like to remind you that my channel is quite small and I'm just getting started. So any form of support, whether it's a comment, like, or a subscription, is highly appreciated and I will strongly value that. So just a reminder, if you guys enjoy my content, please make sure to subscribe, like, and comment. But without further ado, let's get started with the questions. So the first question here, it states, which structure found in eukaryotes has a single membrane? So in eukaryotic cells, we know that there are membrane-bound organelles, but we're looking at something with a single membrane. So we can use the process of elimination. The nucleus has a double membrane. We know that for sure. So this is going to be across. The lysosome, again, this one, I'm pretty sure this one has single membranes. This is our answer, but let's just check option C and D. Chloroplast and mitochondria. You should recall that these structures in eukaryotic cells actually provide evidence for endosymbiotic theory, which is the idea that these membrane-bound organelles came from previous single membrane cells, prokaryotic cells, which were consumed by a larger prokaryote. So the mitochondria and chloroplasts were individual prokaryotes that were consumed by larger prokaryotes and eventually became organelles. So this leaves us with option B being our correct answer. Next question, which evidence falsifies the Davison-Danieli model? So this is the lipid sandwich model, which suggested that the phospholipid bilayer. So this is the phospholipid bilayer surrounded by layers of protein. So it's the lipid sandwich model, the protein sandwich model. The presence of globular proteins within the phospholipid bilayer. Again, since we're trying to disprove this, or what disproved the Davison-Danieli model, this would disprove it because in this model we had two layers of proteins on the outside. This is correct. Non-polar amino acids cause proteins to remain embedded in membranes. That's also true, because that's essentially the same idea. And a further feature of the Davison-Danieli model is that everything's fixed, right? Nowadays, we have the Singer-Nicholson model, which is the fluid mosaic model. And the word fluid means able to move. This one is fixed. So the fact that this statement 3 mentions fixed proteins in a membrane, this actually supports the Davison-Danieli model. So this option is eliminated. So we got option B being our answer. The following electron micrograph shows part of a palisade mesophyll cell, which is labeled structure controls the gas exchange of substances, controls the exchange of substances to and from the cell. So palisade mesophyll cell is just a fancy way of saying plant cell. And IB generally likes to use that term, but just don't be tricked by it. So if we're talking about movement of substances to and from cells, that would be the plasma membrane. This is the cell wall. This is not the plasma membrane because it's literally inside the cell. C seems to be part of the cell wall as well, so A would be our answer. Actually, I'm not sure whether it's A or C because now that I think about it, this could be the plasma membrane here. I think this whole region can be considered the cell membrane, and the fact that this one is more inner and this one's outer might mean that C is the one that controls, just because it's inner versus outer. Outer would have a greater control of movement from inside to outside of the cell, because A would be controlling more to do with this movement, whereas this one would be more to do with this movement. So I'm going to go with C. The diagram shows the concentration of 4 cyclins during the cell cycle. Which curve represents cyclin that promotes the assembly of mitotic spindle? So remember that the G2 phase would be right before mitosis, as expressed here. So whichever one that increases in concentration right at the time of G2 phase or towards the end of G2 phase would be the one that's responsible for mitotic spindle, because mitotic spindle, as the name suggests, is relevant to mitosis. So it can't be A because it's increasing throughout. can't be B because it's already decreased by S phase. C, it might be tempting because it's increasing at G2 phase, but we see it's slowly decreasing before the mitosis phase, so it's not C, leaving D as our answer. And it makes sense because we have this constant increase along towards the mitosis phase and the eventual decrease afterwards. So it makes sense that D is our answer. A polymer of alpha D glucose found in plants has mostly 1,4 linkages and 1,6 linkages. Which molecule fits this description? So, if we're looking at plants, we could have um, two different types. So we can have well, first of all, I need to discuss about amylose and amylopectin are types of starch, right? Because starch is broken down by amylase, and whenever you see this amyl part, that's to do with starch. So this could be starch, but given the linkage, I believe this is going to be something that's. So first of all, we know that it's also branched, right? Because it says that we have 1,4 linkages and 1,6 linkages. So because we have these different linkages, it's going to be branched. So I know that cellulose can't be the answer because there are two different types of linkages. Glycogen cannot be the answer because we're generally dealing with a plant cell. And amylose is not going to be branched in the 1,4, 1,6. So it's going to be amylopectin, which is our answer. This is kind of hard to explain, but I will make a video on the branching. You just have to know that 1,4 linkage and 1,6 linkage refers to amylopectin. Branching is quite complicated in polysaccharides. In an experiment, the effect of changing pH on the enzymatic reaction is tested, which could be a dependent variable in this kind of experiment. Dependent variable, what we measure, and since we're measuring enzymatic activity, what we're measuring should be an effect of an enzyme acting. Changing substrate concentration, that could happen, that could work. Rate of formation of product, sure, that could work. Variation in temperature, no. Changing pH, no. So it's one of these ones, and because we're looking at enzymatic reaction and effect on pH, we're going to be looking at how fast something's happening. So if we're looking at fast, it's better to measure a range. So we're going to go with option B for this. Substrate concentration would decrease like that, but the rate's more important for the pH stuff, because if you've done the experiments before where you have like amylase and how fast that's acting, or hydrogen peroxide and pepsin and those molecules, you've probably done some of that lab experiment in class. It's always a rate that's the dependent variable, because when we measure the enzymatic reaction or the enzymatic activity along with changes in pH, temperature, and substrate concentration, we're looking at the rate of enzymatic activity. So we need a rate as a dependent variable as well. A strand of mRNA consists of the following nucleotides. Okay. Which of the following represents a non-transcribed sense strand of the DNA? Okay. A strand of mRNA consists of the following nucleotides. Which of the following represents the non-transcribed sense strand of DNA? So we have the transcribed strand and the non-transcribed strand, right? So the sense strand is going to be the non-transcribed strand, and the antisense is the one that's transcribed. So AS and S. The easy way to remember is sense makes sense, and that's the one that directly is related to the mRNA. So that literally means this mRNA is the mRNA version or the RNA version of the sense DNA. So we just got to swap out the T's and the U's of the mRNA with the DNA. So we actually don't find something that's complementary. We just swap the U's with T's. That's all we do to get the complementary stand. So it would be A, T, T, C, this one. And there would be option B. What type of molecule is shown in this diagram? So immediately we see the carboxylic region here. And since it's carboxylic region, we're going to think of some type of acid, so fatty acid, and all the options of fatty acid. Here we have a double bond, so it's clearly unsaturated, so we're going to get rid of these. And cis and trans aren't really used for saturated, so these are eliminated. And here we see that the hydrogens are on the same side of the double bond, so that's going to be same, so that's going to be a cis bond, right? So since it's a cis bond, it's going to be a cis unsaturated fatty acid. So we're to determine whenever there's a double bond, they can either be cis or trans. And trans means the hydrogen would be on opposite sides, right? Here they were both on the same, so it's cis. And cis unsaturated fatty acids are actually good for you. They're high in avocados and other substances, other healthy fatty fruits. And B is our answer. Messelson and Stahl conducted experiments using the isotopes and 15 showed that DNA replication is semi-conservative. What would they have observed about the distribution of isotopes in DNA after one round of replication? DNA replication was conservative rather than semi-conservative. Well, DNA half-half, that wouldn't be correct because that would mean it is semi-conservative. Half the DNA with only N14, half the DNA with N15. So what they tried to do was they have this pure DNA with N14, N15, and they moved it to the normal nitrogen environment. And what they saw was the DNA was half N14, half. So do you recall how DNA has two strands? One strand was 14, one strand was 15, and that was the case after the first round. So that means that the original 15 strand is semi-conserved, right? It's half of it's conserved because the original one would have been two 15 strands. One strand is conserved, semi-conserved, for the other strand to be synthesized from N14 DNA, right? So we have an N14 and N15 pairing, so DNA double helix from that. That's why we have a semi-conservative process. If it's conservative, that means the N15s would stay together and the new ones would be N14s. So there'll be N14, N14 pairings and N15, N15 pairings. So half the DNA with only N14 and half the DNA with only N15 would be our answer. So D is our answer. A child has blood group A, father has blood group B. What are the possible genotypes of the mother? So if the father has blood group B and the child has blood group A, that means the father must have been IB, right? And a small I, because if it was IB, then the child could not ever have just an A. He would have either been B or AB. And the mother, so that you need a IA. So the son could be IAI, or it could be IAIA. Now, given that we know that the father is IB I, we know that this child cannot be IAIA, right? But the mother could have passed on the IA gene, right? Because the mother would have had to pass an IA and paired up with the father's I, so that the son or the child, actually, I don't know why I said son, but yeah, so the child would get a phenotype of A, and this is the only combination that could give this is the only genotype of the child. And if we look, well, the only constraint here is that the mother passes on an I, a big I, a allele. And if we look at all of these, they all possess a big IA allele. So we can pick 1, 2, and 3 as any of our options. So D would be our answer. So what proves that the inheritance of the condition shown in the pedigree is autosomal recessive and not autosomal dominant? So whenever you look at dominant or recessive, we got to think about parents and the offspring. So if we have two unaffected parents that produce any affected offspring, we know that effectively tells us that we're looking at autosomal recessive. Because if we recall about recessive, you can have carriers. Carriers are individuals who contain the allele for a disease without actually displaying the phenotype of the disease. And here we have these two guys who are unaffected, or one guy and one girl who are unaffected, and they are carriers for the disease, but they do not actually show the symptoms. So they all so the symptom has to be recessive, because dominant diseases, autosomal dominant diseases, they actually don't display a carrier status. Because if you had one allele for the dominant, that would be expressed in the phenotype. So C would be the best answer. The micrograph shows 4 different phases from meiosis to what is the correct order. So looking at these, we can see 4 different cells, 2 different cells, 2, 2. So this is clearly the last phase. This should be near telophase, so it's one's going to be the last one, and looks like they all consider that. So seeing that 4 has these really uncoiled regions of DNA, I'm thinking that's the first phase, so this should be like prophase 2. So we're going to eliminate options A and option B. So we're going to be left with C and D as our potential options. So looking here, we see 3 is sort of like a metaphase 2 stage where it's aligned along the equator of the cell. So because of that, I'm going to pick C as metaphase 2. So let's just order these. So I'm going to say this is telophase 2, this is prophase 2, this is metaphase 2. And accordingly, we have the chromosomes being pulled apart here. So this will be anaphase 2. So it's going to be, the order is going to be 4, 3, 2, 1. 4, 3, 2, 1, classic football formation. 4, 3, 2, 1 is going to be our answer. So C is going to be the answer. Euglena is a unicellular organism that feeds on bacteria and uses CO2 as a carbon source, which describes the nutrition of this organism. So since it feeds on another living organism that's not autotrophic, we know that it's going to be heterotrophic. But it also uses carbon, CO2, inorganic carbon, right? Carbon dioxide is inorganic as a carbon source. And by definition, an autotroph is an organism that uses inorganic carbon to create organic carbon. Given that it says that carbon source, we can assume that's organic carbon. So from inorganic to organic carbon, it's heterotrophic, but it's also autotrophic. Sorry, I mean, um, CO2 inorganic to organic carbon means autotrophic, not heterotrophic. But because it feeds on bacteria, it's heterotrophic. So it's going to be option D, it's both. This diagram shows a version of the carbon cycle, what is indicated by the numbers. So here we have atmospheric carbon, so this is the carbon sink of the atmosphere. We have tree, and here we can see that atmospheric carbon to tree, that would mean how CO2 from the atmosphere gets into trees, that's photosynthesis. Dead animals to CO2, that should be like decomposition. Living animals to um, coal, oil, and gas, that would probably be like death of consumers leading to fossilization, something like that. So number one, um, first of all, let's look at number 3. Photosynthesis is the answer, so we get rid of these 2 options. Number 2 would be um, incomplete decomposition would be the answer, so both of these are correct. Number 1 would mean, so death of an organism directly wouldn't lead to CO2 formation, but rather cell respiration in saprotrophs, because saprotrophs break down dead organic material, so the dead animals would be broken down by saprotrophs, and saprotrophs, like other living organisms, require cellular respiration, and as a byproduct of cellular respiration, we get carbon dioxide. So C would be the answer to this question. Methanogens produce methane gas. What is the gas converted to in the atmosphere? So if we look at methane, right, this is more something you need to remember off memory. I remember it's converted to carbon dioxide and oxygen. Sorry, carbon dioxide and water, I believe. So if we look, methane is CH4, and when in the atmosphere, it should be converted to, well, I remember in the atmosphere it combines with oxygen mainly, so it can form reaction like this, CO2 plus H2O. I believe it's like this. Yeah, that should be the equation with state symbols. You don't need to remember the equation, just remember carbon dioxide and water formed from methane. A bacterial population with no resistance to an antibiotic may develop into a bacterial population with some resistance to an antibiotic. Which event could lead to this? Antibiotic resistance was inherited from an ancestral population. Inheritance doesn't directly confer to a resistant population, so this is not correct. An antibiotic resistant plasmid is received from a bacterium in another population. Okay, receiving a plasmid could lead to the population having some resistance because plasmid transfer can transfer genetics and cause bacteria to have altered traits, such as resistance to antibiotic. The enzyme needed for antibiotic resistance is received from a bacterium in another population. You can't exchange enzymes really, because you would have to exchange enzymes constantly over and over again. So this is incorrect. The bacterial population mutated in response to antibiotics in the environment. This is a tricky point where students may misinterpret how natural selection and mutations work. Mutations are random and they do not occur in response to environmental constraints or environmental requirements or niches, right? Mutations are random and adaptations are not just happening random. They don't happen in response to something, right? Just because there's a hot day, your body's not gonna adapt and develop like a layer that's free from like a thinner layer of skin. It's not like that. It's over time how small changes and mutations and variation accumulate, right? So you can't just have mutation happening all of a sudden. So according to the process of elimination and because it makes sense, B is our correct answer. An animal shows radial symmetry, has only one opening leading to. Okay, so already because it's radial symmetry, I know that it's going to be a Nidaria. Nidaria is the only one that shows radial symmetry. Annelida does not. These are all bilateral. Cladograms can be created by comparing DNA or protein sequences. Cladogram on the left is based on DNA sequences, and the cladogram on the right is based on comparing protein sequences. Okay, so we see there are quite the same relationships. So we have the same order, but some of the closeness in the cladograms is different. So the amphibians are further than the turtles in the protein. I believe this is the protein. This is protein, and this is the DNA. This one asks, what is the reason that cladograms based on DNA sequences are more reliable predictors of the phylogenetic relationship of species than cladograms based on protein sequences? So when you talk about phylogenetic relationships, that's talking about phylum, and phylum's to do with classification and common ancestors, right? So if something's from the same phylum, that means they share a common ancestor. So it's saying, why do DNA sequences show better, closer relationships than proteins? Amino acids are not as chemically stable as nucleotides. That could be true. DNA mutates, but amino acids do not. That's also sort of valid, but I don't think that's it. Several different triplets of bases can code for the same amino acid. Now, that's a valid point because if you think about it, you can have the same amino acid even if the DNA sequence is altered. When we're thinking about cladograms, right, what matters is the DNA itself because DNA is ultimately how we measure whether things are close to or not, like in terms of a molecular level. We don't care about the proteins. And the DNA is better than the proteins because proteins, as you may recall, are made from amino acids, and amino acids are due to the can be the same amino acid can be coded for by multiple different codons. So even though the DNA may change, leading to a different codon being transcribed and making RNA into a different codon, you can still have the same amino acid synthesized. So so the actual change, the phylogenetic change, or the change, evolutionary change in a species may not actually be detected in the protein, but DNA can actually detect that. So C would be the correct answer. The diagram below shows part of a membrane of a neuron. What stage of the action potential does it depict? So we're looking at the side of neuron, and we see from the outside that sodium plus is moving on to a sodium channel and the potassium plus channel is closed. So potassium channel is open during repolarization, and because it's closed, we know that this is not going to be repolarization. And here we have the sodium potassium pump being closed, so that's not resting potential. So we can eliminate this option as well, and it leaves us with depolarization and hyperpolarization. Now, I already see that from the outside to the inside of the neuron, where we have sodium plus moving down. And because of this, we have generally depolarization, and this won't be a situation of hyperpolarization. Okay, because hyperpolarization means we become even more and more polar. But what happens when sodium plus enters the cell? Is you reduce the polarity. So the inside of a neuron is negative relative to the outside. So this negative pole is how large the negative difference is, right? So if we plot it on like, oops, so since this is negative, how big this difference is. So the polarity is based on how negative this is. So if you have the y-axis here and the x-axis here, how negative this value is how polar it is. But when sodium plus diffuses in down the electrochemical gradient, since this is positive, it makes it less negative. And because it's less negative, the polarity has decreased. So it cannot be hyperpolarization, because hyperpolarization implies the polarity becoming hyper or high. And so the only option is going to be depolarization, which makes sense. Okay, guys, upon reviewing the mark scheme, I have deduced that we got 20 out of 20 for these 20 past paper questions in paper 1. If you guys enjoyed this video and want me to make a longer video where I do maybe 40 past paper questions so that I can cover pretty much a whole paper 1 and do more videos on IB biology past paper questions, please don't forget to smash that like button and leave any comments down below for any series or topics you want me to cover in my channel. And again, a subscription would be highly appreciated, because for me, I only have like 37 subscribers, who I'm grateful for, but um, uh, any little subscription will be very heartfelt for me. So that'll really show your support to the channel. And other than that, guys, this is going to be the end of the video. If you guys enjoyed, again, like, subscribe, comment. And other than that, see you guys in a future video. Peace out.